Well, good afternoon, everyone. Good afternoon, and thank you for joining us. Welcome to the 2025 Don P. Giddens Inaugural Professorial Lecture Series. My name is Ed Schlesinger. I'm the Benjamin T. Rome Dean of the Whiting School of Engineering, and it is truly a pleasure to be here with you to honor Ben Langmead. He is a professor in the Department of Computer Science. He holds a joint appointment in the Department of Biostatistics, and that's at the Bloomberg School of Public Health here at Johns Hopkins. Ben's research focuses on high-performance computing to create open-source software and resources for life scientists. Tools he has developed, tools that he has developed address common genomic research questions that make it easy to query large collections of archived sequencing datasets. He's a member of several professional societies. He's on several editorial boards, and he's received many awards, including teaching awards and an NSF Career Award. And in fact, he received his most recent award about 45 minutes ago when we were over at the Imagine Center, and he received a mentoring award for his service as a mentor. So he's receiving awards on a very accelerated pace. His talk this evening will explore how he built genome indexes as a graduate student that were small enough to fit in memory while still being fast enough to keep pace with DNA sequencers, using the Burrows-Wheeler transform. And this is the foundation for numerous sequence analysis methods. And I have to say that when I was preparing these remarks and I was given some of this information, I looked up what the Burrows-Wheeler transform is. It's actually a pretty clever idea. So Burrows and Wheeler deserve getting their name on it. Well, with soon to be thousands of high-quality human genomes, he'll explain how using pangenomes can improve analyses and how the Burrows-Wheeler transform is still the perfect basis for sequencing data sets. Before we get going, I would like to take a moment to recognize one of my predecessors, Don Giddens, for whom this inaugural professorial lecture series is named. Don Giddens was the 5th Dean of Engineering at Johns Hopkins and a professor of mechanical engineering. And with that, it is my pleasure to introduce Randall Burns, the Bill and Lisa Stromberg Department Head and Professor in the Department of Computer Science. Randall?
Thank you, Ed. I'm here to introduce Stephen Salzberg, which I'll do eventually, but before I do that, I can't not talk about Ben. So I have this tradition in computer science of making some pretty elaborate announcements when there are promotions. And in Ben's case, I was feeling a little harried and, needed to kind of preserve my resources. So I just took an announcement, I threw it in a Burrows-Wheeler transform, and I emailed it to the department, which everyone thought was a puzzle, and ChatGPT 4 at the time couldn't solve it. And Ben, how long did it take you to solve it? Pretty quick. So we have someone that knows about the Burrows-Wheeler transform. I also went on to say, it was truly a pleasure to represent Ben through this process. He continues to do some of the most important work in computational biology. He has defined the software landscape for the study of pangenomics and metagenomics in the period since his last promotion. I'm pretty envious of his approach. He gets to use the coolest data structures, doing things like finding the threshold to detect exact maximal matches using the R index. Ben is an excellent scientist, exemplary member of the department, and a startlingly awesome human being, and that came with an asterisk that I don't need to refine. So Ben joined JHU in 2012, and he was the first faculty member we had in computational biology. And I remember trying to sell him on this endeavor. I believe I was running the hiring committee at the time, and I used some terms that don't sound that great in hindsight, like clean slate or make your mark. And I think this was, I recall being recruited to Hopkins and having the same thing told to me and being suspicious when I was the second member in systems. And he certainly rose to that challenge. Our bioinformatics and biotechnology program is now ranked number 4 in the nation as a discipline of computer science. We were first ranked in 2021. And today, in this forum, I will give Ben all of the credit. And we wouldn't have grown the area without him. He was very active in participating in all of the things you're supposed to do to grow a program, hiring, strategy, recruitment. But I want to focus on like the intangibles. So I think if I was thinking about coming to work at Hopkins in the space, I would say first, I want to work with Ben. And then the second thing I would say is I'd like to work at an institution where people can accomplish the kind of things that Ben Langmead accomplishes. And I think that this is particularly inspirational talking about recruiting and growing. If I was a junior faculty member looking at Ben, well, let me actually say this the other way around. As a junior faculty member looking at Ben's career, I think it's inspirational. And he provides a roadmap and a vision of how to thrive in an institution, really almost singularly. He writes software because he's an engineer, it's fun. He advances computer science theory and data structures. He solves pressing problems in biology and medicine. He collaborates with diverse teams. He's a beautiful teacher. He has a huge impact on the global scientific endeavor, and he does it all with aplomb and joy. For me, this is a cautionary tale. It implies that you can have it all, and personally, I'm not ready for that message, but apparently Ben is. And I also want to comment on Ben's role in advancing the impact and visibility of Hopkins. The biotechnology and bioengineering program is one measure of that. But his open source software is just remarkable. And he has some of his greatest hits include 24,000 citations to Bowtie, 52,000 citations to Bowtie 2, 19,000 to HiSat, and 5,000 to Kraken 2. I don't know that there are 5,000 people on the planet that could read the Kraken 2 paper. I did recently, or I at least looked at the words. But it reflects that what people are doing with Ben's software is they are using it in their own scientific endeavor and citing in that context. And look through this lens, Ben is really impacting the advancements of science through the tools he creates, and that's a lot more than doing science, which he's doing as well. And so on top of this, he's an amazing department citizen, amazing teacher. I mentioned he won 2 teaching awards, but I want to highlight what Ben did for the CS department as chair of the curriculum committee. So I blew that. We asked Ben as an associate professor to overtake the curriculum committee, which is one of our most important functions. And the context was that we had both had a change in our student population and a rise in computer science from the Bloomberg gift. We also had this Q2 mission to increase flexibility in the major. And so I gave this to Ben at the time and said, make this happen. So in the process, he defined all of our current curriculum processes. He also oversaw a redesign of the major to make it more flexible and inclusive. The outcome here that I think is the best is that the CS major now you can enter the degree without any prior training and complete a computer science degree at Hopkins in 4 years. This is inclusivity in a new form, and it's great for our department. Additionally, it's still ABET compliant, and Ben managed all of the social, political, and technical problems of building a major that is flexible and compliant. We have the most flexible major in the Whiting School. So Ben Langmead for president. And with that, I'd like to introduce the Bloomberg Distinguished Professor of Biomedical Engineering and Computer Science and Biostatistics, Steve Salzberg. Steve is also the director of the Center for Computational Biology, is a active and thoughtful science blogger, in addition to being a great scientist, and was Ben's PhD advisor. Welcome, Steve.
Okay, thanks, Randall. So in thinking about what to say about Ben, I tried to come up with an anecdote that would embarrass him, but he was a very kind of well-behaved graduate student, so I don't have any embarrassing stories to tell, but I would like to say a little bit about how we worked together and how he started this project called Bowtie that he'll be telling you about, a descendant of the project. So back in the late 2000s, Ben was a graduate student at Maryland. And he was also working, he kind of went back and forth with a professor at Hopkins Biostats named Rafael Rosari. And at the time, Hopkins had just gotten one of these newfangled machines for DNA sequencing called now we call them next-gen sequencing machines. Actually, now they're starting to just be called sequencing machines, but for a while they were next-gen. And this sequencing machine could sequence DNA incredibly fast compared to everything that come before. So for those who aren't genomics people in the audience, you all have 3 billion base pairs of your genome, of your DNA. These are letters from this 4-letter alphabet in every cell in your body. And every one of you has a slightly different genome. That's why we're all a little different. And when this machine came out, it could generate millions of little short pieces of DNA, not the whole genome, but little short pieces of DNA. And what scientists wanted to do with that, and what they still do today, is they'll take some DNA from a sample, maybe a tumor sample, maybe just a normal healthy person, and they'll get millions of pieces, and they want to compare them to standard reference of like what a healthy genome looks like, and find all the differences. And they'd like to do that quickly. So this machine came out. The first ones were in 2007. And there was some software with the machine, and it was quite slow and cumbersome. And my friend and colleague Rafael Rosari was working with Ben. He said, "We've got some data." We didn't have it at Maryland, which is where we were at the time. And it's this software to do this task called alignment, of aligning these short sequences, we call them reads, to the genome, would take like 2 days for one run of the machine. The machine would also take like a week to run, but then you'd have to wait 2 more days to get the alignment, to find out what are the differences between this DNA and this reference genome. And I thought, oh, we can do that better. So I asked Ben, who is a talented young student, and I had this idea. I said, well, we have this older program that uses a data structure called a suffix tree. And why don't you try modifying that to do this task? And he did that in like a few days, but it still took a lot of memory. And then on his own, he came up with this idea that you already heard Ed mention, to use this completely different data structure called the Burrows Wheeler transform. This came from a different area of computer science, data compression, which if you've ever made a zip file, you've used a data compression program. So it came from a different area, and he thought, well, I could modify that algorithm and data structure to do this other task in genomics, to align sequences to a genome and do it really fast and do it in really little memory. And he came up with the first version of Bowtie, implementing that. The only credit that I think I can claim for that is that the name Bowtie was my suggestion. Because we needed a B, W, and T in order in a single word. And so I guess there's one other small credit, which was my older daughter designed the original logo for that. And my older daughter, who was also a babysitter for Ben's kids a long time ago. So anyway, he designed this program. It worked on a laptop, so it used far less memory than any of the other programs that were out there. And I still remember, and I'll just conclude on this because this was on social media, and I can't remember if it was Twitter because this was like 2008, but on social media at the time, people, when he first released the code, someone commented that they started their alignment run of their data, and they expected it to take 2 days, because that's usually what it took, about 48 hours. And they went to get coffee, and they came back 20 minutes later, and it was done. And they assumed the program had crashed, because there's no way it could be done in 20 minutes. But it was literally close to 100 times faster than some of the nearest competitors. So that was kind of the beginning of Bowtie, and that's why the program, as you heard from Randall, that's why the papers describing this program have tens of thousands of citations and tens of thousands of scientific users around the world today. So it was a delight to work with Ben. It was great to have a student who you basically I could just say, like, why don't you look at this problem? And he would go off and come up with this very creative idea to merge ideas from 2 different areas of computer science and produce something that everybody really needed and had this huge impact. And I've been delighted to watch his career and see him get all his teaching awards. And I've used his teaching materials myself in teaching. He's better at presenting these ideas than I am. So I'll let him now try to explain to you how you use some of the latest ideas for pangenomes. Ben Langmead.
All right. Thank you. Thank you so much. I've commented to multiple people that seeing this particular room of people assembled is something that before today would only have happened in a strange dream that I was having. So it is good to be with all of you here today, and it's the real life. So I'm here to tell you about two lines of work, one of them hearkening back to what you were hearing about what I was working on in graduate school, but then to also reemphasize why this is actually still, in fact, in some ways even more so, a really excellent line of work to be pursuing in the current environment. And I want to add two little provisos at the beginning. So first of all, some of what I'm going to be describing is going to be ideas from the computer science literature that weren't my ideas. So for example, the Burrows-Wheeler transform is something I'll describe today. Other people did that work. I'll try to be clear when I'm talking about our work. But then also when I say our work, I want to be clear that I don't just mean me. I mean a wonderful lab and a wonderful set of collaborators. And on this particular slide, I'm only showing PhD students, postdocs, and software engineers who've worked with me. I have a much larger thank you slide later on, but I just wanted to make sure you understand that when I say we, this is who I mean, we. Also, as was the advice that I was given, I'm going to stay at a pretty nice, broad, explanatory, demystifying level for today. But if you are intrigued and you want to learn more and you have lots of free time, you can go to my YouTube channel, and I have far more videos than you will want to watch there. But I'll probably have some that address what you wanted to hear about. And I also have them on Figshare if you don't want to watch ads. Okay. So I am a computer scientist, and I work on algorithms for analyzing sequencing data. So what is that? Why do computer scientists work on that? What do I really mean? Well, I want to set a frame for today's talk, exemplified by these two things, these two sort of moments in time. So one of them, the one on the left, is the Human Genome Project. So if you were reading the news circa 2000 to 2003, you'll remember hearing about the completion of the draft sequence of the Human Genome Project. You might remember press conferences with Bill Clinton and Tony Blair and people like that. This was a project that produced the first draft sequence of the human genome. It's called a draft. It wasn't perfect, but it was very useful as a reference for future projects. I'll describe what I mean by that. The second set of milestones I have there are much more recent. So one of them was just a few years ago, the T2T. T2T basically means end-to-end, complete, gapless human genome assembly. This one fixed most of the flaws with the draft human genome assembly from the Human Genome Project. And then the second is the Human Pangenome Reference Consortium. So this is a group of people whose mission is to assemble as many high-quality human genomes as possible. And they've assembled upwards of 200 of them so far. Okay, so I show these two projects not because I worked on them, though I do slightly work on the Human Pangenome Reference Consortium. I do work with them. But mainly because they frame today's two topics. You know, why has it been important so far, and what makes it important for the future of these topics? So let's start with the Human Genome Project. The Human Genome Project, as I say, was the project that produced the draft sequence, the first draft sequence of the human genome. Genomes are made of DNA. DNAs are A's, C's, G's, and T's. I think many of us sort of know this already. A for adenine, C for cytosine, G for guanine, T for thymine. So when that project assembled the draft sequence of the human genome, what they assembled was a sequence of A's, C's, G's, and T's that was 3 billion letters long. Billion. So one amusing way to represent this and sort of visualize how much data this is the exhibit that you see on the left, which was a real museum exhibit in London. I don't think it's there anymore. But it actually had a printed out version of the human reference genome sequence in a bunch of bound volumes. So that's what you're seeing there. Those are the chromosome numbers on the spines. There's one book open on the shelf right there. I think I have a pointer. On the shelf right there. If you were to zoom in on that book, it's literally a wall of single-spaced, ACs, Gs, and Ts. Okay, so that gives you some sort of physical idea of just how incredibly massive this project was, right? It produced a sequence of 3 billion letters. That's that many letters, but on every page of every book you see on that bookshelf. Okay, that project did something else too, which is it proved to biotech companies that if they could build a better sequencer, they could make a lot of money. Because the Human Genome Project, this was a 13-year, $3 billion project. It's usually what people estimate it at. But the sequencing alone was on the order of hundreds of millions of dollars. So it sort of proved to biotech companies, hey, maybe we could do a better job. Maybe we could make sequencers that are cheaper, faster, and more convenient. And so many companies got to this task, and they wildly succeeded, and Stephen already mentioned this. After the Human Genome Project, circa 2007, there started to be products hitting the market that we would now call second-generation sequencers. At the time, they were called next-generation sequencers. And these were hugely, vastly less expensive than the sequencers that were used for the Human Genome Project. This curve is showing survey data from a survey that was done by NIH, asking people, hey, how much does it cost you to sequence 1 human genome's worth of DNA? And what you're seeing from 2007 to 2011 is a 3 order of magnitude plus drop in the cost. Okay, huge drop. So that says part of it, but I think what says much more of it is the fact that we now have, as normal products on the market that we can be prescribed by our doctors, things like sequencing-based diagnostics. So here's a test, for example, that you can have done. It's called the Foundation 1 CDX test. It helps home in on what's the right treatment for a particular person's cancer. It's a sequencing-based, it's a next-generation sequencing-based product. And not only is it FDA approved, this is an approval, it's also, believe it or not, covered by insurance. So it's even covered by Medicare. So just as an example, I think biomedical people would see something that's FDA approved and covered by Medicare as a pretty wild success. So this is a good example of how next-generation sequencing has made it into our everyday medicine, and it goes beyond medicine too. So here's a small selection of examples. This one's about forensics. This one's about evolutionary biology. This one is about the surveillance that happened during the COVID epidemic and how sequencing was important to tracking which strains were propagating. This one's about an idea to use DNA for data storage. So you would use sequencer as essentially like the read head of a storage device. Okay, so sequencing. It really became far more convenient and widely used after the Human Genome Project. But how does it work? Like, what does it do? Okay, back to our DNA here. Ideally, what we would love for a sequencer to do is we'd like to give it a sample. Maybe the sample is a blood sample or something like that. That sample has cells. The cells have DNA. We'd like to extract the DNA. And then we'd love for the sequencer to just read from one end to the other, like an entire one of your chromosomes, for example. So maybe we would just read it off like that. It can't do this, or at least it can't do it very well. The reality is that sequencers are pretty good at reading short stretches with high accuracy or long stretches with relatively low accuracy, but you sort of can't have both. You can't have a sequencer that reads very long stretches at very high accuracy, not possible. This is, here's an analogy would be, for example, I can read, I'm happy to report to you, but if you were to give me a book, a long book, and say, Ben, please read this book from beginning to end without stopping or making any mistakes, I wouldn't be able to do that, right? I would have a coughing fit, or I'd lose my voice, or something else would happen. Similar limitations for sequencers. They cannot read long sequences with high fidelity, okay? But what they can do really, really well, and I'm focusing on this part right here, is read many, many short sequences with very high accuracy. So let's look at a cartoon picture of this. Let's say we're sequencing a sample from you, and this is one of your chromosomes. The sequencer, what it's doing is it's essentially reading off randomly selected little sections of the DNA. Okay, many, 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 many of them. And so this is what we started with. This is what the sequencer has reported. At the top, we would call those reads, just a bit of vocabulary. So everything that the sequencer reports is called a read. So those are our reads up there. And the thing to know about the reads is they're actually incredibly short relative to the chromosome. So for example, a typical human chromosome is on the order of 100 million letters long, A's, G's, and T's, but one of these sequencing reads is only on the order of 100 or 200 or so letters long. So these are so short that they're really not at all edifying to look at one at a time. They don't help you. Like they're not even long enough to span a typical gene, for example. So what we absolutely have to do first with a computer, so what we have to do with algorithms first, is we have to take these reads, we have to imagine that they are puzzle pieces, and we need to put the puzzle back together so that we can see the more complete picture. Again, the individual reads, not helpful. The completed puzzle, that's actually something that can help us study what's going on with this patient, for example. Okay. And you know what really helps if you're putting together a puzzle is it helps if you can see the picture of the completed puzzle on the lid of the box. I think we've all done that, right? So if this is the picture of the completed puzzle on the lid of the box, combine that with the pieces, we're off to the races because what we can do is, I mean, in this example, I think what I would do is take out all those red pieces that look like they come from that nice red flower and set them aside, and I already have kind of a head start on the overall puzzle, right? So your general strategy could be take a piece, hold it up to the picture on the box lid, and use that to figure out where to put it. That might sound a little circular at first, right? Because these are the pieces of my genome. So how do I know what my completed genome is before I've put together the pieces of my own genome? Well, that's where the Human Genome Project and genetic relatedness can both help us. Because if you look at two unrelated humans, their genomes are extremely similar, 99.6% or so similar by sequence. So if this is my genome here on the left, and this is Shohei Ohtani's genome here on the right, those two genomes would be about 99.6% similar. Similarly, the genome that was assembled as part of the Human Genome Project also would be 99.6% similar to my genome. So it's true that I don't really have a picture of my own completed puzzle to use as a reference, but I have a picture of someone else's completed puzzle that I can use as a reference, and that is what we call good enough. Asterisk, good enough asterisk. Okay, and I'll come back to the asterisk later. So we can use the genome of somebody else as a guide for putting together, say, my own puzzle, my own genomic puzzle. Okay, so when I got to grad school, as Stephen did a great job of introducing, the burning question was, how do you come up with algorithms and data structures that are able to do this, and to do it at a speed that actually can keep up with the speed of a typical sequencer? Okay. That animation will look better on Zoom, I hope. Okay, so now for the fun part. Right now we can talk about algorithms and data structures for analyzing sequencing data. Okay, first thing I would say is if we're going to work with this data, we would love it if all of the data that we're working with fit in memory. Okay, so Stephen also alluded to this. It's a little bit like if you lose your car keys, but you're sure they're in your bedroom. They won't be too hard to find, right? But if you lose your car keys and you have no idea, they could be in your bedroom, they could be anywhere in your house, they could be in your car, they could be in your office, they could be in your parents' attic, you have a lot of work to do. Similarly, we would like all our data structures to fit in memory, to be in our bedroom, so that we can get this done as quickly as possible. These days, typical memories are like, let's say 16 billion bytes or more, 16 gigabytes or more. Back when I was a grad student, not so lucky, my computer had 2 billion bytes or 2 gigabytes available. So this was sort of the budget that we were working with. Let's see if we can cram everything into 2 gigabytes. Okay, does the human genome fit in 2 gigabytes? Well, I said that it was 3 billion letters long. So if each of the letters required a byte, the answer would be no. That would exceed our budget. Luckily, each of the letters of the genome is only one of 4 choices, A, C, G, or T. That means we only need 2 bits to distinguish A, C, G, or T, and a byte is 8 bits, so we can pack 4 of them into a byte. Okay, so we can pack 4 letters into a byte, do the division. That means it takes 750 megabytes, so fantastic. That does fit in memory with room to spare. So far, so good. Okay, however, that's not exactly the data that we want to be looking at in order to solve this problem. Right? That's the picture of the completed puzzle, great. But we don't want to have to hold a puzzle piece up to the picture of the completed puzzle and try 3 look in 3 billion separate places to see, is this the right spot? 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 That's not going to work. That's going to take far too much time. It's a little bit like if you imagine using a search engine, right? When you put your query into the search engine and you hit enter, you get like a near instantaneous response back from the search engine. That's not because the search engine was, in that interval of time, painstakingly searching every byte of the internet for your query. It's because the search engine has a data structure back there called an index. And that's what we need here too. We need an index. That's what will make this solvable. Okay. Index. Like the index of a book, perfectly good analogy for the sort of index we would like to have. If you wanted to look up a word in this index of a book, like let's say you wanted to look up mutilation, ritual. There's a book about insects, okay? So you wanted to look up that word, no problem. You know how to do that because the index is in alphabetical order. We all internalized alphabetical order early in life. We know how to look something up very quickly. A computer can also do this very quickly. So a computer would follow a set of instructions you might call binary search. It would take the thing you're looking for, it would compare it to whatever is the middle word, the word right smack in the middle of the index, and ask, okay, is what I'm looking for before or after this? And if the answer is before, then you can ignore the entire second half of the index, throw it in the trash, you're not going to use that, just recurse on the first half. Do the same thing, but for the first half. That would take a logarithmic number of steps because that's repeated division. You're dividing in half each time. So just like it's convenient for us to use the index of a book because we know how alphabetical order works, we can do something similar for genomes. However, we can't directly apply that same idea because there's just no notion of words here, right? There's no, punctuation or spaces or commas or sentences or anything like that. It's really just a constant stream of letters, right? The joke about studying sequences is it's just one thing after another. So somehow we have to build an index of this that respects the fact that whatever it is we're looking at, it could start anywhere, right? We can't just decide that this is a bunch of words because what we're looking for could start at any particular letter. We need a kind of letter-oriented index that understands every single letter. So that is what we would call a full text index from the title of the talk. So we need to build something called a full text index. Okay, how do we do this? Well, I'm going to suggest something, and it's going to seem weird. It's going to seem a little strange. But if we start with a text that we would like to index, let's say it's this text down here, what we're going to do is we're going to take the letters of that text and put them in a new order. Okay, same letters, but different order. We're just going to rearrange them. But this new order is going to be the best order, the best possible order that we could possibly put them in if our goal is to use it as an index. Let's see what that order would be. So as an example, let me just pick a couple letters out of this string. Okay, pick another letter out of this string. Okay, there we go. Hopefully you can see that the two letters are blue, right? The A and the C, I'm putting in blue. And then their context, so the stuff that comes immediately after them, that's in red. So you can see the context of this A is CTT, that's what comes directly after. The context of this C is CTA, that's what comes directly after. We're going to compare those two contexts alphabetically, and if we do, using usual alphabetical order, what we're all familiar with, and if we do that, which one comes first? The CTA comes first, right? CTA comes before CTT. So in that case, what that means is that the letter that has the context CTA, that's this C, is going to come before the letter that has the context CTT in our rearrangement. So because CTT is greater than CTA, that means this A is going to be after this C in our new arrangement. Okay, so much for A and C. Let's pick this C right here instead. There we go. So I just moved over to that other C, right? This C has a different context. Its context is TAC. So if I compare CTT to TAC, well, CTT comes first. So in the case of this C, the A is going to come before this C. Okay, so now we know a little bit about what the new order is going to be. It's going to have this C before this A, before this C. And let me just go ahead and do this for every single letter. Let's see how this animation looks. That was pretty good. So there we go. This blue string here, okay, which by the way, it respects those two decisions we made earlier. Here's the C and A and C that we were just talking about, and it's in the order that we said. Now we have all the letters of the original string in order according to their context. Okay, believe it or not, this is it, right? We did it. So now you understand the Burrows-Wheeler transform, okay? So all it is a rearrangement of the letters, except for that this arrangement of the letters is in some sense the best. I'll explain that a little bit more. And you could also, I've been using the term rearrangement, we could also use the more mathy sounding permutation, right? It's a permutation. We just put the letters in a new order. We permuted them. Okay, this permutation here, which is a permutation of that there, is so useful that it has the fancy name that we've been talking about this whole time, the Burrows-Wheeler transform. It was first proposed by two British computer scientists, though they were working in the US at the time, Michael Burrows and David Wheeler. They were at Digital Equipment Corporation in their research department. So they proposed this. I'll say more about what they proposed it for. Actually, Stephen said it already, it's for compression. But what was pointed out later, and which is also amazing, is that this particular permutation is also useful as an index. And this was pointed out in the year 2000 in a paper by these two computer scientists, Paolo Ferragina and Giovanni Mancini. Okay. I'm going to explain now why it's useful as an index, and this is a good place to remember that I have a YouTube channel, because I'm going to go pretty quick and use pictures. Okay, so I'm going to go quickly and use pictures, but if you are more curious, if you feel like I'm glossing over what you're interested in, youtube.com/Ben Langmead. Okay, so here's the picture we made before. I'm going to modify it in a couple of ways. The first thing I'm going to do is turn it into a matrix where the contexts are still here, but they're expressed as what are called rotations instead of the immediate right context. And then I'm going to take that thing that we called the Burrows-Wheeler transform that's in blue, I'm just going to shift it over to the right-hand side. And I'm going to concentrate mostly on the first and last columns. Okay, so I've sort of grayed out everything that's in the middle. It's not so important for what we're going to talk about. You might be a little bothered by the fact that I just sprinkled a bunch of dollar signs in there that might look weird. That's a sort of boring technicality, no particular need to explain that now. So here we go. We've got our column here, which we know, that's what we said before. That's the Burrows-Wheeler transform. Those are those blue letters we made before. These letters here, you can think of these as their most immediate right context. These are the most immediate contexts of these letters in the original string. A relationship exists between these two columns, the red one and the blue one. It's called the LF mapping. And it can be drawn like this. I just drew a bunch of lines connecting letters from the first column to letters from the last column. And this relationship, the LF mapping, is exactly what allows us to navigate this thing and actually use it as an index. Let me explain this in the form of an example and a picture. Okay, so let me go to a query string. Let's say that we want to figure out all the places in our original string where this pattern occurs, TAC. We're going to go through 3 steps now, one for each letter of our pattern. We're actually going to start at the right-hand side, so we're going to start with the C. And we're just going to travel through this data structure according to those green lines that I put on the screen on the previous slide, one by one. Okay, so let's start with the C. Okay, good. The animation turned out pretty well. So that's the first step. Let's go on to the A. That's the second step. Let's go on to the C, and that's the third step. We were following those usual green lines that I put before that I called the LF mapping. And where we land at the end of those 3 steps is exactly here. So here's our pattern TAC. And we landed right here in these 2 rows because these are the 2 rows that begin with TAC. So these are the 2 instances where TAC occurs in the text. So by following that procedure, we figured out, yes, TAC is present. It's present 2 places. Here they are, right? Just from those 3 steps. Okay? Importantly, each of those steps happened in basically an instance. So we can do this, computer scientists would say constant time. So just as quickly as you could imagine. So snap, snap, snap. Those 3 snaps were as quick as it took to match that pattern to that text, that particular database. It doesn't matter how big this thing is, right? This is a really small example. It fits on my slide. But if I were to move to another example where the index is the index over 3 billion letters from the human genome, or 3 trillion letters from 1,000 human genomes, it wouldn't really matter as far as how fast this algorithm is. This algorithm would go in 7 snaps because my pattern is 7 letters long. It does not depend on how long this thing here is, right? It's not 3 billion snaps, it's just 7. So this is the first huge success that we get when we get the Burrows-Wheeler, when we use the Burrows-Wheeler transform as an index. Notice we're not doing any painstaking scanning through the entire genome. That'd be far too slow. We're not even doing the binary search thing that had logarithmic steps, also would be too slow. It's just instantaneous for every letter of the pattern that we're looking for. Okay, to add another victory on top of this one, it's also gonna fit in our memory budget. So let's go back to our memory budget picture. We already said the genome fits with room to spare, great. What is the Burrows-Wheeler transform? It's just a permuted version of the genome. So it's exactly the same size as the genome. It's all the same letters, they're just in a different order. So it's the same size, so it also fits. That adds up to 1.5 gigabytes if you're doing the math. So far so good, we still fit in 2 gigabytes. And then there's some more stuff I'm not gonna talk about. Again, YouTube, if you're interested in knowing what that other stuff is, but just imagine that it's the stuff that makes it fast to walk across those green lines that I showed you in the previous diagram. All of it fits in our 2 gigabyte budget. So even back in the year 2007, 2008, this data structure could function very, very quickly as an index of the queryable index of the human genome, and it could fit in 2 gigabytes. Okay, so this discovery, which is what Stephen was talking about earlier in the introduction, propelled many, many years of work that wasn't just limited to grad school. It was also spilling over into many years of my professor career, because once you build a piece of software that people start using, you've got more work to do, because if you want them to keep using it, you have to keep making it better and fixing bugs and responding to their questions and making it have good documentation. All of this, by the way, is open source software, so all of this is free, easy to use. The source code is out there for anyone to look at. And so several years, in several years, starting from the original paper describing Bowtie, we did a lot of other work to build on that, make it better and better. Stephen correctly mentioned that he was the person who gave me who called it Bowtie the first time. And he said, isn't that a great name? It's got BWT in it. And I thought, I can do better. So I got the dictionary and I tried to find other words that had BWT in them, and the best ones I found were browbeat and bewitch. So we went with Bowtie, and Bowtie is a fantastic name. I'm very glad. Thank you, Stephen. Okay, so that takes us to partway through the professorial journey, but the title of my talk is From Strength to Strength, or that's what's in the title of the talk. And I've told you really only about the first one, right? I only really told you about why these ideas are appropriate for, say, the human genome that came from the Human Genome Project. Now I've got more to say about what's coming in the future, because this is the part I'm actually quite excited about. Okay, so let me go back to these two projects that we mentioned here on the right-hand side. These two projects are both examples of why these days the sequencing technology has become so good that we can not only sequence short sequences of DNA with high accuracy, we can now, these days, sequence longer stretches of DNA with pretty high accuracy, like okay accuracy. And that is the key to being able to make new things like the human genome, but better. So new things like the draft sequence of the human genome, but more accurate and contiguous and covering the entire genome without any holes or mistakes. Much could be said about these technologies. They're very, very cool. This is one example. This one, which is called smart sequencing, is basically, there's a tiny well. In the bottom of the tiny well, they've set up the sequence polymerization reaction, the reaction that makes copies of DNA. And then it's being observed through a tiny, tiny little pinhole camera. And you can actually sort of observe the bases as they're being integrated and sequence it that way. There's another technology called nanopore sequencing that works by taking the DNA, drawing it through a pore, very small pore, nanopore, and then measuring at each moment the degree to which the DNA is blocking the flow of ions through the pore. And that's an indirect way of measuring which bases are in the pore. These are both now pretty highly developed and good working technologies, and they're what are allowing this part of the space to be populated with effective tools. In fact, it's kind of like, I didn't make an animation here, but just imagine it's kind of poking slowly up further to this not yet achievable corner of the diagram, these technologies. What they can already do for us, and what they've already done for us, is let us build a much better human genome sequence. So I mentioned this project, the T2T project, the gapless human genome project. I mentioned earlier that the Human Genome Project produced a draft sequence that had flaws. Well, this one didn't have any flaws. This one was actually quite close to perfect. Okay, maybe there were just like 5 places left that they couldn't quite sequence across, but still, to be polite, we usually just say they did it. You know, it's completely gapless. And indeed, this picture here is actually a diagram showing all the gaps that were in the original draft human genome sequence, but that they filled in this new sequencing effort. So everything that's red here is an example of something that was basically missing for many, many years from the references that we were all using to study you and other people. Okay, and this was a project led by Karen Miga and friend from graduate school, Adam Philippi. And it was a project that included many, many other people, including Mike Schatz, who's sitting right there, and others here at Johns Hopkins, including Johns Hopkins students. Okay, and so in 2022, we got our first truly gapless human genome assembly. The Human Pangenome Project, which I've mentioned a couple times now, is an effort to say 1 is great, hundreds would be better. And so this is an effort to sequence and assemble literally hundreds, maybe one day thousands of human genome reference assemblies to a very high quality. And you'll see, this is a diagram from their first paper, they're casting the net wide, right? Like this is not a project to sequence, like let's say a bunch of Europeans, right? This is a sequence to project a very wide array of people from around the world so that we have a reference that includes genetic diversity, right, from different parts of the world. Okay, it's up to 233 humans. This data is available. There's not yet a publication describing it. But the reason why I'm so excited about projects like this is because remember back when I said that comparing data from one genome to that of another person's genome was good enough, asterisk. I now want to return to the asterisk and say, remember I said 99.6, right? Unrelated people are about 99.6% similar by sequence. There's a deficit there of 0.4%. That 0.4% is not just a nuisance, right? Like this isn't just a little bit of randomly distributed noise that separates one genome from another. This actually is concentrated in certain places, and it actually makes it impossible to study certain things, this remaining variation. So for example, one region of the genome that you could point to is called the major histocompatibility complex, or MHC. It contains immunity-related genes, important genes. But for good evolutionary reasons, this part of the genome is what would be called hypervariable. So it's quite different from person to person. So instead of being 99.6% similar between unrelated people, it's closer to 95% similar between two unrelated people. Still, you know, it's in the 90s, that sounds good, but it's a lot lower than 99.6. And at that point, using this as a reference for someone else fails to be a good idea. It stops being a good idea. This is a plot that some people in this audience will recognize as an IGV plot. What it's showing you is a puzzle that has not been put together very well. And that's because this reference was not a good representation of the genetics of the person who was actually sequenced. They were too distant. And now it's a little bit like gibberish. It's not very helpful. This is just one way that this can manifest. We take all the different ways that you can make mistakes when you use a reference genome in analysis of sequencing data, and we put a great big term on all those mistakes, call it reference bias. Reference bias comes from the fact that the person you're sequencing is actually different from the reference, and because they're different from the reference will sometimes drive you to make certain mistakes. Systematic mistakes, right? That region that I showed you, we will always make mistakes in that particular region, right? Again, it's not randomly distributed across the genome. It also interferes with a lot of very specific things people would like to study, like allele-specific effects. Or sometimes they want to filter host DNA from a sample that has both human and other critters' DNA, and they want to get all the human out. In those examples, if you use a single reference genome, like the one from the Human Genome Project, you systematically make mistakes. This has also even been written about in the popular press. Here's an article from Stat that's referencing the fact that the individual who was sequenced primarily in the Human Genome Project was from the Buffalo area. So that's why it's saying Buffalo gave us spicy wings in the Book of Life. It's basically saying not a good idea to keep just using this one person's genome as the be-all and end-all reference for all our work. Okay, but I'm here to tell you, right, there are solutions available. So for example, instead of using as our reference the human genome, what if we were to use the human pangenome? This is one genome. This is hundreds of genomes. Why would we do that? Why would that sounds harder. Why would we do that? Well, because if we can give ourselves 100 chances to find an individual who's similar to us in a certain region, then chances are we're going to find a hit. We're going to find someone whose picture on the box lid is a good match for our puzzle pieces. But we have to give ourselves those many opportunities first. We have to have a pangenome representation rather than a genome representation. So for example, let's say there's my genome again, and let's say there's a region in my genome, hopefully you can see the red, where there's some genetic variants there. And let's say these genetic variants are actually very important to my health and well-being, but they're rare. So a typical other person won't have these same genetic variants. And let's say this is the reference genome from the Human Genome Project, and it does not share those variants. It is different from me in this region. Okay, so if this was the only chance we gave ourselves to study my genome, was to use this as our guide, as our template, as the picture on the box lid, we might make a mistake in exactly the place where it counts. However, if we had a pangenome representation of several people, that say these people are all sequenced as part of the Human Genome Project, there's a pretty good chance that somewhere in there, there's a hit. Right? There's somebody who's got the same genetic pattern as I do. And if we can use that genome as the picture on the box lid, we're going to do a much better job of assembling that very important part of my genome. So that's the goal for the future. The single biggest remaining problem, I would argue, that we have with the accuracy of sequencing data analysis is reference bias. This is how we can address reference bias with many genome sequences. Now, I'm using the term pangenome. You might, if you go out and explore the literature, see other terms that are similar but not exactly the same, like reference panels, or like personalization, or imputation. These are all really different ideas in the same bucket of using many genomes to provide a good reference for studying one new one. But this, as computer scientists, we computer scientists have a new problem to solve. We have to build indexes that are over hundreds of genomes instead of one. That sounds hard, right? Remember we struggled to, or okay, maybe it didn't seem like we were struggling, but it was hard to get in under that 2 GB budget with our single human reference genome. So you might think, well, now we need 200 GB or something like that. But the Burrows-Wheeler transform has got this, right? This is what the Burrows-Wheeler transform came to do, was to help us with this problem. So back to the Burrows-Wheeler transform, and Stephen mentioned this in his remarks. It was originally invented for compression. And in fact, you probably haven't used it for this, but there was a tool, somewhat popular. It was called bzip. So different from zip and different from gzip. It was called bzip. And it was based on this idea. It was based on the Burrows-Wheeler transform. Didn't quite make it as a popular compression tool. However, it's obviously very popular as an indexing tool. But the whole point was to do compression. And the observation that they are making in their paper is that if you do put letters in this new order, according to the alphabetical order of their contexts, what you tend to get is a repetitive sequence in a given window. So let me show you an example. And so let's look at a pangenome together and see this idea in action. Here we go. So here's my ultra realistic, real-world pangenome. Okay. It's just a toy example, but sometimes it's easier to see when the example is in English because the patterns really jump out at you. So here's a string, and let's say a sequence, and we'd like to index it just like we were using the Burrows-Wheeler transform before. We'd like to use it here too. And what you'll notice, of course, is that this is repetitive. In fact, there's almost like 2 dimensions of repetitiveness here. Right? There's the fact that the 3 lines are repeats of each other. So there's the vertical kind of repetitiveness. But then also within a line, row is repeated. So there's a kind of horizontal repetitiveness as well. So there's sort of 2 different versions. So what will happen if we apply the Burrows-Wheeler transform? Well, when we take letters and we put them in that new order according to their context, what will happen is that letters that tend to have the same context also tend to be the same letter. So letters that have the same context tend to be the same exact letter. So for example, let's highlight the W's here. Those are all the W's. All the W's have pretty similar stuff after them, right? So these W's have space and then R, and these W's also have space and then R, and then these W's have space and then Y, which is pretty close to R. So these W's, when we apply the Burrows-Wheeler transform, they're going to come together. They're going to be sort of attracted to each other and clump together in the Burrows-Wheeler transform. Okay, So let's do it. Let's look at the Burrows-Wheeler transform of this string. Right away, what you can observe is there they are. Those are all the W's clumped together, just like I said. But also this clumping has happened all over the place, right? Like this is a very clumpy permutation. It's got lots of what we call runs of the same letter repeated. So the Burrows-Wheeler transform took the kinds of repetitiveness that were sort of inherent and latent in the original input, and it made it really obvious and brought it to the surface, which is very useful if we'd like to compress it or index it. In fact, if we wanted to represent this in a compressed way, an easy way would be, instead of just writing these repeated letters over and over again, O, 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 let's save ourselves some time and effort just by saying W times 9, for example, for these 9 W's in a row. So if we represent it this way, this is called run length encoding, then we end up with 15 runs to describe what was originally 64 letters. Okay, so we got 64 letters down to 15 runs. And roughly speaking, that's like a compression ratio. It's a bit like we started with 64 things, now we've got 15. Do the division, that's a compression ratio of about 4.27, which is remarkable because if you look back at the original example, if we had only been able to take advantage of one kind of repetitiveness, like let's call it the vertical kind, we probably would have got 3 as our compression ratio, but we got something greater than 3, which tells us that we took advantage of both the vertical and the horizontal kinds of repetitiveness here. This kind of repetitiveness, this is a toy example, but this kind of repetitiveness is something we do expect when we have human pangenomes. Because, for one thing, 2 unrelated humans are 99.6% similar, as we said. So that's the sort of vertical kind. Also, any given human genome is also quite repetitive. There was even a period of time you might have heard the term junk DNA before we knew what all the DNA in the genome was useful for. It is not fashionable to call it junk DNA anymore, but the DNA that was referred to as junk was stuff that looked like this, very repetitive. So even within a human genome, there's this horizontal kind of repetitiveness that you see here. Okay, so much for row, row, row your boat. I want to show you that this works for humans, that this works for human pangenomes. So let's see. If we actually use the Burrows-Wheeler transform on a large collection of human genomes, high quality human genome assemblies, and this is the absolute best set that we have right now. It's from the year 2025, like it's a recent data set. This is 466 haplotypes. I said earlier it was 233 people, but each person has 2 copies of all the chromosomes. So that's why this is that times 2. So we have 466 haplotypes. And if we think of each of the haplotypes, as I said before, as giving us another sort of swing at the pitch of being able to observe the genetic variants that you truly have in your genome, if each of these haplotypes is one more swing, then what you're seeing is that we're getting 466 swings, which is to say 466 human haplotypes, for a cost that's only about 1.58 the cost of a single string, so only a single swing, so only about 50% more expensive than what we would have needed in order to store 1 human genome. So as a compression ratio, we can store 466 haplotypes in about 550x compression ratio, so we can compress it down by a massive amount. This is the key, right? So this is why the Burrows-Wheeler transform, it's not just something that was great at the dawn of the next generation sequencing revolution, it's also the key to being able to index pangenomes so that we give ourselves every possible chance to have a good reference for the person that we're trying to assemble, but for only a marginal cost, only a tiny, tiny little cost. Okay, there's lots of other work that's really all about taking the Burrows-Wheeler transform based indexes. I showed you two different permutations earlier, right? The Burrows-Wheeler transform itself is a permutation, so that was the first one. Then the LF mapping was another one. That's another permutation. There are others too, and I won't bore you with all the details, but the process of doing pattern matching using these indexes uses a variety of them, a variety of these different permutations. Everything ultimately is made of permutations in this data structure. So what the previous slide was basically saying is that one of those permutations has incredibly good scaling, right? It scales what we would call sublinearly. So even though we multiplied by 466 the number of haplotypes, the actual space usage only went up by a factor of about 1.6. The other permutations at play, including the LF mapping that I mentioned before, which I drew as a bunch of individual lines on a previous slide, also respects this new scaling factor, right? Because even though the LF mapping still maps each of these letters to each of these letters, The LF mapping does it in a highly redundant way, whereby all the letters that are part of the same run are moving in parallel through this permutation. So it becomes something more like a block permutation. We're permuting chunks of text instead of individual letters. That too has the BWT run scaling that we desire. So the things that make it small and the things that make it fast are all scaling favorably, according to that same function I showed on the previous slide. So this has given rise to a whole series of software and papers. It's even a little confusing, I think, sometimes for people. What are all these ideas? Well, even though the Burrows-Wheeler transform seems like a simple idea, it's at the basis of a lot of different tools that are out there. So it's at the basis of tools that treat a pangenome as a bunch of sequences, which is sort of what I've been talking about here. It's also at the basis of a lot of tools that try to take all the sequences and compose them into a graph, a certain kind of diagram, and then index that graph. And there's also tools that try to very quickly take a quick peek at your data and then infer the best possible reference genome for you in a personalized way. All of them use the Burrows-Wheeler transform. And so there's a lot of different lines of work that emanate from the Burrows-Wheeler transform, but that are trying to make pangenomics a very practical reality. Okay, getting close to the end. So I showed you before the Foundation CDX test. Many, many inputs went into this, right? The fact that we can do this affordably and quickly with next-gen sequencing, and now it's FDA approved and it's covered by Medicare. One of those paths, I would say, is the one that went through the computational breakthroughs that made it possible to analyze sequencing data from them so that we don't worry too much about whether the computational part of analyzing this sample is going to be the bottleneck. You know, whether what we're waiting for is the computer, or whether what's the most expensive part is the computer or the computational time. We just don't worry about that. And I would say a big part of why we don't worry about that is because there's been a highly successful interplay over many years between theory and practice. And so one example is the storyline that I was saying today. It started back with Burrows and Wheeler, who were working at a company, Digital Equipment Corporation, but in their research division. So this is like an industry-academia kind of hybrid situation here. This is academia, this is academia, this is industry. It's also an interplay between theory and practice. This is theory, this is theory. This is a transition from theory to practice. This is sort of theory and practice getting married, and then practice. And so if we're going to make a similar trip into the future, I would argue that we're following essentially the same path. And we can arrive using things like the Burrows-Wheeler transform and other ideas in a world where sequencing-based analyses are efficient, cheap, and effective, regardless of your genetics, right? You don't have to look like the reference genome to get a good answer back from your assay, which is good because that's what's going to make it effective for every part of the genome, not just the easy parts, and for people of every genetic background. Okay, with that, I'm going to return to my first thank you slide. This again is my PhD students and postdocs and software engineers. Thank you very much. And now here's the one that was really hard to do. The final one where I had to think of many, many people and where I'm kind of terrified that I'm missing some people. But I especially want to thank two people who were instrumental in sort of introducing me to and guiding me through the recent literature on pangenome indexing, which are Christina Boucher, who's at University of Florida, and Travis Gagey, who's at Dalhousie University in Canada. They've been a huge part of my research agenda since about 2018 or so. And thanks to many more wonderful colleagues and collaborators, including ones I couldn't list here. Thank you.
Any questions for Ben?
My in-laws are here, so easy ones only, please.
How does the human mind work? I mean, it's an incredible group of structures, and we take it for granted that we'll just remember everything.
Yeah.
And cognition, of course, declines as we get older. So how does, briefly, how does that compare to this? And then I'm going to ask you about AI.
Okay, all right, I'm ready. Yeah, there's a number of researchers here who can do a much better job than I can to speak to things about the mind. I have worked with some neuroscientists, so I have worked with people who do use sequencing and genetics to study the brain. And usually what they're studying is neurodegenerative diseases that have some kind of genetic foundation, because that's the kind of thing that sequencers can help you to study, right? You can start to figure out Is the mutation everywhere? Is it from birth? Is it just in certain tissues, et cetera, et cetera? But the mind, in terms of how it works dynamically in any given moment, you need to measure that different ways other than with DNA sequencing. You need to go in there with fMRI machines and other things like that to try to study what's happening live. Genetics won't really necessarily help you do that, though there's some branches, things like transcriptomics and spatial transcriptomics, which I don't study, but that can be used to study things like that. So mostly what I've talked about today is useful for studying the genetic basis of diseases, but not necessarily for what's happening live in a person's brain.
I'm considering it on an index level.
Mm-hmm. Yeah.
How does it come to a conclusion
Yeah.
and make you aware of it
Yeah.
so fast? And of course, AI is
Yeah.
better at it, even though
Yeah.
it has its own faults.
Yeah, AI can do some of these same things. So for example, if I was mentioning search engines as something where you get an answer back instantaneously. One of the things you'll notice search engines can do for you is they can actually finish your question for you, right? You can start asking your question and it'll finish the word before you finish typing the word. So you might say, oh, it must have a full text index back there, right? If it's finishing my word, but it's not, it's an AI model, like you're saying. That's what's really behind the scenes. But that's a situation where what it's trying to do is make a good suggestion. It's not crucial that it give you the exact right suggestion, right? And so the sort of lossiness of an AI model where it doesn't actually know exact answers to a lot of questions, it just sort of knows a good way to intuit an answer to a question, that won't necessarily answer the same questions that we're trying to answer with these data structures. There is a field called learned data structures. So this is sort of like data structures that can be improved by using AI to, for example, predict what the next question is going to be, and then try to reconfigure what the data structure is to answer that question better.
I have found
Yeah.
that using different AI, as you use one, it becomes more familiar,
Uh-huh.
of course.
Yep.
And
Right.
it leads you on and it guesses better and
Yeah.
better.
Yeah.
Like a person.
Right.
Or like,
So, yeah, one example of where people have tried to bring those ideas together in areas like this is this learned data structures. They're trying to learn how you're using the data structure so that they can reconfigure the data structure that's better for the sorts of queries you're asking. So that is something people study. Yeah. Yeah. Yeah, yeah. Absolutely. Yeah, yeah. Yeah, I didn't say it, but back here I wanted to mention that, you know, the fact that we don't worry too much about how much the computation is costing or how much time it's taking, that's in contrast to AI, right? We're in an AI world right now where there's a conversation about, should we be committing these resources, the land and the data centers and the electricity and the water, right? This is something we don't really worry about here, but I would argue that's in large part because of work that people have done to make sure that this computation is never the bottleneck.
One more question.
Sure. Yeah.
Did you ever think what you would have done if you only had one device?
Given up. I would have given up. Yeah, it's a good question. In fact, I'm glossing a little bit when I say that it was everything fit in 2 gigabytes, because the reality is we needed 2 indexes, the one of the forward version and the one of the reverse version of the genome. So the reality is we had to come up with an intricate dance to schedule the computation so that only one of those could be in memory at the same time. So if you gave me 1 gigabyte, more complicated dance, but maybe we could do it.
Well, I think it's clear why you wouldn't teach anymore. Thank you very much. And
Go ahead.
a small token of Professor Langmead's first inaugural professorial lecture.
Thank you.
Join me here.
Thank you.
Congratulations.
Thank you.
And please join us for refreshments.